What if the deadliest creatures on Earth held the key to saving millions of human lives? While most people fear venomous snakes and cross streets to avoid them, scientists have discovered that their lethal toxins contain miraculous healing properties that revolutionize modern medicine. In Thailand alone, over 2 million venomous snakes are systematically milked each year across 47 specialized facilities, producing 380 kilograms of pure venom that becomes life-saving anti-venom, groundbreaking cancer treatments, revolutionary blood clot medications, and advanced pain management solutions. The Queen Saovabha Memorial Institute in Bangkok houses over 18,000 deadly cobras, Russell's vipers, many banded kraits, and Malayan pit vipers in a sprawling facility that resembles a pharmaceutical laboratory more than a traditional snake farm. Here, highly trained herpetologists wearing specialized protective equipment extract venom from creatures that can kill a human in less than 15 minutes. Yet paradoxically, this same venom saves over 185 snakebite victims annually across Southeast Asia and produces medications used by 65 million patients worldwide. This extraordinary contradiction challenges everything we think we know about nature's most feared and misunderstood predators. The irony runs deeper. While Hollywood movies portray snakes as villains, these same creatures provide compounds that treat heart disease, stroke, chronic pain, and even show promise against aggressive cancers. Join us as we dive into the fascinating, dangerous, and ultimately life-affirming world of venom extraction, where death literally becomes medicine, and humanity's greatest fears transform into our most powerful healing tools. The revolutionary practice of snake venom extraction began in 1895 when French scientist Albert Calmet made a groundbreaking discovery at the Pasteur Institute. While studying cobra bites in French Indochina, Calmet found that small controlled doses of cobra venom could immunize laboratory animals against larger, potentially fatal doses. This eureka moment launched what would become a multi-billion-dollar global industry that produces anti-venoms, cardiovascular medications, neurological treatments, and cutting-edge cancer therapies. Today's snake venom contains over 200 different proteins, enzymes, and bioactive compounds, each with unique and often multiple medical applications that pharmaceutical companies are still discovering. Compounds originally found in Brazilian jararaca, viper venom revolutionized blood pressure treatment and now help 85 million hypertension patients worldwide through ACE inhibitor medications. Meanwhile, cobra neurotoxins show extraordinary promise in treating chronic pain conditions without the addiction risks associated with traditional opioids. The global antivenom market alone generates $2.8 billion annually, while venom-derived pharmaceuticals contribute an additional $4.2 billion to the healthcare economy. Specialized extraction facilities operate across 6 continents, with major operations in Thailand, Brazil, Australia, India, South Africa, and the United States, each maintaining massive snake colonies specifically designed for sustainable venom production. These high-security facilities house everything from deadly black mambas and fer-de-lance vipers to tiny but lethal coral snakes, with each species contributing different therapeutic compounds to humanity's medical arsenal. A single gram of dried king cobra venom commands prices up to $45,000 on pharmaceutical markets, making it more valuable than gold, platinum, or even some precious diamonds by weight. The extraction process demands extraordinary expertise and courage, as trained handlers work within striking distance of creatures capable of delivering fatal envenomation through fangs that inject venom faster than a human can react. Creating a world-class venom extraction facility begins with meticulous selection of snake species based on their specific medical applications and venom potency profiles. Facilities prioritize highly venomous species like king cobras for neurological research, Russell's vipers for blood coagulation studies, many banded kraits for pain management compounds, gaboon vipers for cardiovascular applications, and fer-de-lance snakes for anti-cancer research. Wild-caught breeding pairs from certified suppliers establish founding colonies through carefully managed reproduction programs, with detailed genetic tracking ensuring diverse bloodlines and optimal venom characteristics. Offspring receive intensive care from birth, spending their critical first 8 months in climate-controlled nurseries, maintained at precisely 78 to 82 degrees Fahrenheit, with humidity levels between 65–75% to prevent respiratory infections and promote healthy development. During this crucial period, specialized herpetologists monitor each snake's growth rates, feeding responses, temperament, and early venom development through daily visual inspections and weekly physical examinations. Juvenile snakes receive appropriately sized prey items every 5 to 7 days, with meal sizes calculated based on individual body weight, species requirements, and growth targets established through decades of research. Advanced environmental control systems maintain perfect conditions through redundant heating, cooling, and humidity regulation equipment, as even minor temperature fluctuations can stress developing snakes and permanently affect their venom production capabilities. The facility maintains comprehensive digital records, tracking each snake's genetic lineage, parentage, birth date, feeding history, health status, behavioral observations, and projected venom yield potential. Specialized ventilation systems filter air continuously through HEPA filtration, while maintaining optimal humidity levels and air circulation patterns that prevent the bacterial and fungal infections commonly found in captive reptile environments. Multi-layered security measures include biometric access controls, 24-hour surveillance systems, emergency lockdown protocols, and immediate medical response capabilities staffed by paramedics trained specifically in exotic envenomation treatment. Comprehensive training programs require new handlers to complete 6 months of intensive education, covering snake biology, behavioral recognition, emergency response procedures, first aid protocols, and graduated hands-on experience with non-venomous species before advancing to work with dangerous animals. As snakes reach sexual maturity between 18–36 months depending on species, their venom production increases exponentially, with adult king cobras capable of producing up to 12 milliliters of highly potent venom per extraction session, while smaller but equally deadly species, like many-banded kraits, yield 0.8 to 1.5 milliliters of concentrated neurotoxic compounds. Adult feeding schedules transition to bi-weekly intervals for most species, with meal sizes carefully calculated using established formulas that account for body weight, metabolic rate, seasonal activity patterns, and optimal body condition scores that maximize venom quality without compromising snake health. State-of-the-art monitoring systems track individual snake behavior patterns, feeding responses, shed cycles, breeding readiness, and venom regeneration rates through sophisticated computer databases that analyze trends and predict optimal extraction timing. Specialized veterinary staff with advanced training in reptile medicine conduct monthly comprehensive health assessments, screening for internal and external parasites, respiratory infections, mouth rot, scale diseases, injuries, or behavioral changes that could indicate illness or stress affecting venom production capabilities. Environmental enrichment programs provide species-appropriate hiding spots, climbing structures for arboreal species, substrate variety, and seasonal lighting changes that promote natural behaviors, while maintaining strict safety protocols required when working with deadly animals. Advanced climate control systems simulate natural seasonal temperature and humidity variations that trigger breeding behaviors, optimize venom gland development, and maintain the biological rhythms essential for consistent high-quality venom production throughout the year. Water quality monitoring systems ensure all drinking sources remain free from harmful bacteria, chemicals, or contaminants that could affect snake health, alter venom composition, or introduce variables that compromise pharmaceutical applications. Selective breeding programs utilize genetic testing to identify bloodlines with superior venom yields, enhanced potency levels, and desirable behavioral traits, allowing facilities to continuously improve their colonies while maintaining genetic diversity essential for long-term sustainability. Experienced staff members develop detailed knowledge of individual snake personalities, learning each animal's unique behavioral cues, stress indicators, feeding preferences, and temperament patterns that indicate optimal health and readiness for safe venom extraction procedures. Venom extraction procedures occur every 14-28 days, depending on species, individual recovery rates, and seasonal factors, with timing carefully calculated to maximize yield while ensuring complete venom gland regeneration and snake welfare. The sophisticated milking process begins with expert handlers using specialized restraint equipment, including custom-designed tubes, adjustable clamps, and protective barriers that safely position snakes while allowing natural striking behaviors essential for complete venom evacuation. King cobras and other large species require heavy-duty restraint systems with reinforced construction, while smaller but equally dangerous species like coral snakes and death adders utilize modified collection apparatus designed specifically for their unique fang structures and striking patterns. Master handlers with decades of experience position each snake with millimeter precision, ensuring optimal fang placement over sterile collection vessels while minimizing stress indicators and injury risks to both the valuable animals and irreplaceable human expertise. Fresh venom appears as clear to pale yellow liquid, with viscosity varying by species, immediately beginning complex protein degradation processes unless preserved through ultra-rapid, flash freezing techniques using liquid nitrogen or specialized cryogenic equipment. The global venom market operates on strict quality grades, with pharmaceutical-grade king cobra venom commanding prices up to $48,000 per gram. Research quality material bringing $18,000 down $25,000 per gram, and educational specimens selling for $5,000, $8,000 per gram, depending on purity and certification levels. Advanced processing facilities immediately stabilize fresh venom samples at temperatures below 80 degrees Celsius, then employ sophisticated lyophilization processes that remove moisture content through freeze-drying, while preserving delicate protein structures essential for medical applications. Rigorous quality control protocols analyze each batch using mass spectrometry, high-performance liquid chromatography, and bioassay testing to verify potency levels, identify contamination, and ensure consistency meeting pharmaceutical industry standards. International distribution networks ship processed venom globally under strict temperature-controlled conditions, using specialized containers, real-time monitoring systems, and chain-of-custody documentation required by regulatory agencies in destination countries. Different snake species produce dramatically distinct venom profiles. Neurotoxic venoms from cobras, mambas, and kraits specifically target nervous system function, while hemotoxic venoms from vipers, rattlesnakes, and adders affect blood coagulation, tissue integrity, and cardiovascular function, with some species producing complex combinations requiring separate processing protocols. The global snake venom extraction industry directly employs over 15,800 specialized workers across 185 facilities in 23 countries, generating combined revenues exceeding $4.7 billion annually through anti-venom production, pharmaceutical development, medical research, and therapeutic applications that benefit millions of patients worldwide. Rural communities throughout Thailand, Brazil, India, and parts of Africa have successfully transformed traditional snake-catching knowledge into highly lucrative, modern careers, with master venom extractors earning between $52,000 to $78,000 annually, plus comprehensive health insurance, retirement benefits, and specialized training opportunities. The global anti-venom market serves approximately 2 million snakebite victims each year, reducing fatality rates from historical levels above 25% to under 4% in regions with adequate treatment access and proper medical infrastructure. Export revenues from venom-derived products contribute substantially to developing nations' economies, with Thailand generating over $220 million annually, Brazil contributing $185 million, and India adding $95 million from venom-based medical products shipped to 89 countries across 6 continents. Major pharmaceutical corporations, including Pfizer, Novartis, and Bristol-Myers Squibb, rely heavily on venom compounds for developing breakthrough medications. ACE inhibitors derived from Brazilian viper venom currently treat over 95 million hypertension patients globally, while experimental cancer treatments using cone snail toxins show remarkable success in Phase 3 clinical trials for treating pancreatic and lung cancers. The rapidly expanding venom extraction industry confronts increasingly complex ethical questions about animal welfare, with wildlife protection advocates arguing that maintaining thousands of venomous snakes in commercial captivity prioritizes human medical needs over individual creature comfort and natural behavioral expression. Industry defenders respond that modern extraction facilities provide superior veterinary care, consistent nutrition, protection from predators, and stable environments compared to wild habitats where snakes face habitat destruction, climate change, pollution, and systematic persecution by fearful humans. Environmental scientists raised legitimate concerns about potential genetic bottlenecking in captive breeding populations and the ecological impact of removing prime breeding adults from wild ecosystems already stressed by human encroachment, deforestation, and agricultural expansion. Worker safety remains the industry's paramount concern, as even the most experienced handlers face constant envenomation risks that could result in permanent disability, career-ending injuries, or death. Despite comprehensive safety protocols, advanced protective equipment, and immediate medical response capabilities, the industry's fundamental dependence on living animals raises philosophical questions about sustainable alternatives, though synthetic venom production using recombinant DNA technology remains at least 2 decades away from matching the complexity and therapeutic effectiveness of natural compounds produced by millions of years of evolution. Global distribution inequality means life-saving antivenoms remain financially and logistically unavailable in many remote rural areas where venomous snake bites occur most frequently, highlighting persistent healthcare disparities between developed and developing nations. As medical applications for venom compounds continue expanding into treatments for Alzheimer's disease, multiple sclerosis, and autoimmune disorders, modern society must carefully balance tremendous healing potential against legitimate animal welfare concerns, environmental impacts, and questions of justice in medical access. What moral responsibility do we bear toward these remarkable creatures that literally sacrifice their freedom and natural behaviors to provide compounds that save human lives and alleviate suffering? Should the undeniable medical benefits justify large-scale captive breeding programs, or can advancing technology eventually eliminate our dependence on living snakes while preserving the healing power of their venom? The answers we choose may ultimately determine whether this extraordinary industry represents humanity's most ingenious adaptation of nature's deadliest biological weapons into powerful healing tools, or simply another troubling example of exploiting vulnerable animal populations for human benefit, regardless of the ethical costs involved. What's your perspective on this delicate balance between human medical needs and animal rights in our modern world?